Okay, hello everybody. My name is Anna Klindworth. I'm from the Max Planck Institute in Bremen. Yeah, I try to speed up, and yeah, I'm part of Frank Oliver's group, and as Renzo already told you, I won't talk about metadata, but I will present you my work about the evaluation of primer and also primer pairs for 16S ribosomal RNA biodiversity studies. And as a majority of you might know, primer play quite an important role within culture-independent studies if those culture-independent studies contain a PCR step, as for example in the full cycle ribosomal RNA approach. And as you can see here, the cycle always starts with the sampling procedure and nucleic acid extraction, which is in most cases DNA, and then it's followed by amplification of your gene of interest, which is in most cases 16 or 18S. And for the amplification, you need universal primers. And those primers are usually designed or chosen based on all the sequences available in the databases. And although PCR is in general quite a, let's say, the simple step, and pretty much every lab can do it and you don't need specific or really fancy equipment, you should never underestimate the step, because choosing the correct primer is actually one of the crucial steps. I mean, if you pick the wrong primers, for example, this could lead to the preferential amplification of certain groups, or worst case scenario, your important key players might be not detected at all. So always keep in mind that this is quite an important step and you should never underestimate it. Then next thing will be that you can use your amplicons for a classical clone library approach, or use next generation sequencing technologies to directly sequence your amplicons. And then in the end, all the sequences are submitted to the databases like SILVA or RDP, and the results can then be used for phylogenetic analysis or probe design. And with each cycle, more and more sequences become available, and in particular with the advent of all the next generation sequencing technologies, the amount of data hardly or increased a lot, as you can see here on the diagram. And it's nice to know that the diversity is intensively studied. However, the problem is that the majority of primers have been designed years ago. Today, some of the primers are even in use which have been designed in the late 90s, and some other primers have been, I don't know, designed 2004. And if you have a look at the amount of data which was here at 2004, I mean, you can see it really caused a backlog, and primer evaluation is crucial, and we try to address this problem by performing an in silico evaluation of commonly known 16S primers. And in the beginning, the first thing we did was actually to do an intensive literature survey to find the most commonly used primer and also primer pairs. And in addition, the community members of the ARB and SILVA community provided us with some other suggestions. And soon it became actually clear that there are several primers out there which contain the same sequence, so we decided to rename all our primers according previously published nomenclature by Alm and colleagues in 1996. And one example can be seen here. It's quite a long name, but it contains lots of information. This would be just an example. S stands for the small subunit of the ribosome. D and also the 4 letters here contain information about the target group. In this case, it will be D for domain and BAC for bacteria. This is the 5' start position of the primer based on the E. coli nomenclature. B would be just like the second primer which has been published, starting at that position. S would be forward primer, and the 17 is the length of the primer. And after we've done this whole renaming process, we ended up with 175 primer, and they were used for the evaluation, which was based on the SILVA non-redundant reference data set. And we were in particular interested in getting the overall coverage for all 3 domains of life. And since the focus of this study was on archaea and bacteria, we also analyzed the phylum spectrum for those 2 domains, meaning that we had a look at each phylum if this contains high coverage, and if, I don't know, using if we use certain primers and which might lead to failure detection of certain members. And since it's known that a standard PCR can tolerate up to one mismatch, we repeated the whole evaluation with one mismatch. And this first step can be seen as a screening tool so that we can exclude the primer with low coverage, so that in the end, only primers with an overall coverage above 75% for either bacteria or archaea were used for primer pair analysis. And as you can see, we ended up with quite a lot of suggestions in total, and we had 512 primer pairs, and they were divided into 3 different groups according to amplicon lengths in group S, M, and L. And the reason why we decided them into different groups, we wanted to provide the scientific community with recommendation for certain sequencing technologies like Illumina, for example. And what we did, we had a look at all those primer pairs, checked the overall coverage as well as the phylum spectrum, and in the end, we provided the community with recommendation for each group. And since I don't have much time today, I only want to show you one example from the group M. And this was actually the best primer pair for amplifying bacteria 16S sequences. And as you can see, it generates almost 500 base pair amplicons. It covers hypervariable regions 3 till 4, and our evaluation revealed that this is a highly domain-specific primer pair. It covers almost more than 86% of the sequences, and unfortunately, this primer pair doesn't achieve a full phylum spectrum. Detailed analysis revealed that 7 bacterial phyla might not be detected. That means that the coverage for those phyla was below 50%, and one example would be Chloroflexi or candidate division OP11. And then the one mismatch evaluation revealed that this primer pair might detect up to 95% of the sequences. However, a small amount of archaea sequences might be detected as well. And what we also did, we had a look at the primer at the mismatch position, because in 3 cases, the mismatch position was close to the 3' end, so we believe that the amplification of those three phyla might be difficult. And what we also did, we did a field study within the MIMAS project as a proof of principle, and we used this primer pair and applied it to marine environmental samples from Helgoland Roads in the North Sea. And the amplicons were sequenced with 454 Pyrus sequencing, and in the end, the results were compared to the 16S sequence from PCR unbiased metagenome samples, and those metagenome samples were generated within quite an interesting comprehensive multi-omics studies. And if you would like to have some more information, I can only recommend that you have a look at this paper, which has just been published in Science. Okay, just a few results. As you can see from the pyrotext, the bacterial community was dominated by 3 bacterial. Oh, okay, I have to hurry up. The bacterial community was dominated by those 3 bacterial groups, and a similar picture could be observed in the metagenome. And I'm going to skip this slide due to time problems. So in silico, in summary, we evaluated 175 primers and 512 primer pairs, and the whole study is intended to serve as a guideline to provide the scientific community with the currently best primer pair for different sequencing technologies, and all the details about the evaluation can be found in that paper, which has just been published in Nucleic Acid Research. And last but not least, all primer information are now available at ProBased. And the Silva now provides also an online service which is called Test Prime, and this allows you to perform your own primer pair evaluation with your primer pairs of interest. And that will be just an example from the ProBased page. And as you can see here, you have your new name, sequence, the target gene, and interesting is that this page is directly linked to the Silva Test Prime tool, and if you click, you can choose your reverse primer, and then you can just click on the button and you're directed to the Test Prime probe, which allows you to perform your own primer pair evaluation. You can choose between the different database, mismatch position, and then you just run your evaluation and you end up with some nice results. Okay, thank you for your attention. I'm sorry this is a bit long. So
time for questions.
So what, this is very interesting. What opportunities are there to go back over the legacy sequence data from all these anticons and annotate those with things you have in ProBase, prime pairs in ProBase?
Sorry, I couldn't.
So what can we do to go back to the legacy data, so all of the sequences we have from previous studies, and annotate those with the prime pairs that you now know about and described in ProBase?
I mean, that's what we did. We used all the available sequences in our reference data set, and then we had a look which was the best primer pair.
Typically, the